Hello and welcome to another episode of CellVec Conversations, where we explore various aspects of CellVec and the wider viral vector manufacturing industry at large. In this episode, we will take a look at what goes on behind the research and development function at CellVec and meet the person at the wheel. I would like to warmly welcome Dr. Paula Lam, Head of Research and Development here at CellVec. Dr. Paula Lam is a bioscience industry veteran, and her research involves exploring various strategies to increase the capacity for viral vector production and establish in-house assays for the functional characterization of these viral vectors in various immune cells. Hi, Dr. Paula. It is great to have you here with us, and I'm sure many people will wonder, what do you do in your day-to-day role?
Hello, thank you so much for inviting me here. From day to day, I oversee the development of projects, research and development projects, the manufacturing process that is scalable and cost efficient while ensuring a consistent and good safety profile. I have a very small team, and each day we plan. So the day will start by integrating the various research activities and among both internal and external meetings, sometimes both in the same day. So it's a continuous drive to identify key scientific gaps in knowledge that applies and is relevant to what we do so that we can improve the overall efficiency and consistency of our research.
I imagine every day is very busy and fulfilling for you and the team. Can you share with us some of the research projects you've been working on?
Sure. Some of the key research activities include the development of a lentiviral manufacturing process that is free of products of animal origin. So this will enable the scale up to a much larger volume while minimizing the regulatory risk associated with animal transmittable diseases, thereby making it more suitable for clinical manufacturing of therapeutic products. So at the same time, this reduces the batch-to-batch variation coming from the animal products, such as from the fetal bovine serum that is used in our manufacturing process, as well as the associated high cost. We can also reduce the cost of production. So taken together, we aim to increase the product yield purity while reduce the cost of manufacturing without compromising the quality of the product. We also strive to continuously improve the upstream and downstream process design to increase the overall lentiviral production.
Now for a slightly personal question, what sparked your interest in cancer research?
Sure. So cancer is actually very complex. It's driven by both genetic and epigenetic alterations that allow cancers to overproliferate, and cancer develop also escape mechanisms so that normally control their survival and migrations. So many of these alterations actually map to very complicated signaling pathway that control cell growth and division, cell damage and repair, cell metabolism, cell death, and these are all factors that can lead to cancer progressions. So I personally found this to be very fascinating, especially intrigued by the 1889 hypothesis by Stephen Paget, where the seed and soil theory. So basically, he states that the seed, which is the cancer cells, is influenced by many factors, such as accessibility to nutrients through proximity to blood vessels, and they need to strive in the right soil or the right tumor environment. So till this date, scientists all over the world are still working very hard towards understanding the intricate network of cancer initiation and progression. So the declaration of war on cancer by Richard Nixon in 1971 is still an ongoing battle. So that made me stay in the field of cancer research due to the fascinating science and the potential impact at addressing a global health challenge.
Oh wow, that's quite interesting. Can you share with us one of your most memorable research projects?
Yeah, to provide an example for a more recent project that I find it also very interesting is at present 70% of the viral products are manufactured using an adherent-based platform where the cells are grown as monolayer in culture vessels instead of free-floating in cell culture medium. So the adherent-based manufacturing process can be readily procure off the shelf and require less expert bioengineering and know-how compared to the three-dimensional platforms. So, however, most of the adherent process are dependent on products of animal origin, such as the fetal bovine serum that I talked about earlier, and this poses safety issues, consistency, and regulatory challenge. So typically, in the absence of serum, the cells will ball up. That will impact on the transfection efficiency and the lentiviral production is therefore poor. We have identified an improved method where even in the absence of serum, we are able to culture a human monolayer of cells to facilitate a good production of lentiviral vectors.
And what are some of your favorite breakthroughs in cancer research?
There have been many breakthroughs in cancer research, and one of my favorites is CAR T cell therapy. The acronym CAR T represents chimeric antigen receptor T cell therapy. In essence, it involved the genetic modification of the patient's immune cells with chimeric antigen receptors that allows functionally distinct T cell subset to recognize specific tumor cells. The incorporation of co-stimulatory molecules or cytokines can enable engineered T cells to eliminate tumor cells. I remember it was one of the scientific conference in 2013 where the clinical results of CAR T therapy were shared. I was totally mesmerized by the contrasting PET-CT images scan before and after infusion of CAR T cells. The patient had entered a phase of complete remission without additional treatment. So I remember distinctly that this was the first thing I shared with a clinician friend of mine upon return to Singapore, that we should look into this innovative therapy for our patients. So Emily Whitehead was the first patient to receive CAR T therapy. The parents were informed by the physician at the time that she has only a couple of weeks to live and it's best to send her to a hospice. So her father learned through a nurse that there was an innovative therapy called CAR T and subjected her to the trial. And even though cancer is a very complex and challenging disease to treat, but it is not impossible because till today, Emily Whitehead is still very much alive. She has grown into a very beautiful young lady and is able to perform her day-to-day activity as a happy teenager.
We really can't wait to see more of the possibilities that CAR T can bring, and we know just how critical CellVec's work must be in advancing CAR T therapy. So, in your opinion, what does the future of cancer treatments hold?
I think the future of cancer treatment is quite promising, especially given the help from advanced technologies such as artificial intelligence and nanotechnologies. We are potentially able to uh, improve early detection of cancer, learn from past clinical experiences, the genetic and biomarkers that are responsible for tumor cell elimination and tumor cell recurrence. There are novel personalized methods for targeted treatment, immunotherapy using conventional small molecules or antibody drugs together with multiplex engineered cells that have begun to deliver curative treatment for cancers. And there are also many next generation immunotherapies that are coming our way. So we have already seen some of the success story of CAR T at treating certain types of cancer. and it is the combination of the immune cells from the patient with the chimeric antigen receptor delivered through lentiviral vectors, which is the heart of what CellVec does, that makes the treatment of cancer, especially in the case of Emily Whitehead, that makes it possible. So I think the future of cancer treatment is quite promising.
It's really been an eye-opening experience listening to your sharing today. Thank you so much for your time, Dr. Paula. Stay tuned for the next episode of CellVec Conversations.